Hello everyone, and welcome to our first lecture video for the Anatomy and Physiology 2 lab class. The first topic that we will be covering in this course are the general senses. Now, general senses was chapter 16 and was covered in Anatomy and Physiology 1. However, those of you that have not taken Anatomy and Physiology 1 for quite some time or can't remember what it is that you did last semester, fear not. I will be going over all of the information that you will need for this lab class. For the quizzes and the practical exams, you want to make sure that you're studying what's on the PowerPoint slides, what's in the lab manual sheets, and any extra information that I say in the class. You'll find all of the PowerPoint slides and the lab manual sheets posted on Canvas under the Modules section. So having already covered general senses in Anatomy and Physiology 1, you should be fairly familiar with some of the concepts that will be presented in this lecture video. General senses as a topic refers to how our body generally experiences different types of stimuli. We must remember the difference between the terms sensation and perception. A sensation is any type of registered change in our body's internal or external environment. And perception refers to our own conscious interpretation of that sensation. This is how two individuals can have different pain thresholds. The sensation may be the exact same, but it is the perception that is different. General senses can be broken down to our body's receptors, and we have many different types of receptors that will function for our general senses. So in this presentation, we're going to talk about a few of the types of receptors that our body has, and what type of stimuli is monitored by each. Receptors can be classified in many different ways. The type of stimulus that the receptor is detecting is one way that we can classify receptors. That's where we get the term mechanoreceptors, thermoreceptors, photoreceptors, and nociceptors. We can also classify receptors structurally based on the physical components that those receptors have. Some receptors will have a connective tissue capsule, and we call those encapsulated receptors, while other receptors are simply free dendrites, and we call those free nerve endings. Individual cells on their own can also act as receptors. However, the way that we are classifying receptors on this slide is based upon the location of the receptors and where the origin of the activating stimuli comes from. This will probably be one of the more straightforward ways that we can classify receptors. So specifically, we have exteroceptors, interoceptors, and proprioceptors. Three different types of receptors we're going to deal with today. Exteroceptors, as they are nicely named, will monitor our body's external environment. In terms of the general senses, touch, pressure, vibration, temperature, and pain are all stimuli that are conveyed through exteroceptors. Those are all stimuli that do come from the external environment. So therefore, we classify the receptors that monitor those stimuli as exteroceptors. Interoceptors, as you can probably imagine, monitor the internal environment of our body. We also call these visceroceptors because they will monitor the health of our visceral organs inside of the body. The amount of stretch in blood vessels and muscles, and concentrations of different chemicals in certain parts of the body, are types of stimuli that are going to be monitored by interoceptors, or visceroceptors. To compare exteroceptors to interoceptors, I'll use real-life examples that we will see in this presentation. An example of an exteroceptor is the Pacinian corpuscle that can be found in the skin. We're going to go over this receptor specifically in just a couple slides, but I want to just mention that the Pacinian corpuscle will sense deep touch. It's found in the skin and it senses very, very deep pressure. So that is a receptor that is monitoring some type of external stimulus. Deep pressure in the skin is most likely going to be caused by some type of external subject. And it is because these exteroceptors monitor the external environment that they will actually be found very close to the external environment also. An example of an interoceptor, on the other hand, is our baroreceptors. Baroreceptors are a very specialized interoceptor that monitor the stretch of blood vessels. They're going to help regulate our blood pressure, and therefore they're sensing a part of our body's internal environment. So that would be one example of an interoceptor. Baroreceptors will be covered in much more detail when you get to the cardiovascular system. Proprioceptors are another type of receptor that we have in our bodies. These are located in muscles, tendons, joints, as well as in the inner ear. These types of receptors give us information about our body's position. The particular muscle length, or perhaps the tension in our muscles, are two types of stimuli that will be sensed by proprioceptors. The proprioceptors in the inner ear, which we will discuss in more detail when we get to the special senses, will also give us information about our body's position and also help with balance. So we're going to go on and elaborate on a couple types of exteroceptors that we have in the skin. Two tactile receptors that we will specifically find in the layer of the skin known as the dermis are the Meissner's corpuscles and the Pacinian corpuscles. Tactile receptors are touch receptors. Tactile means touch, so therefore these receptors are going to sense mechanical stimuli around the skin of the body. Meissner's corpuscles and Pacinian corpuscles are both examples of tactile receptors. However, they sense different types of touch, as we can see on this slide. Meissner's corpuscles specifically will sense light touch, and they're found just nestled under the dermal papillae, while Pacinian corpuscles will sense deep pressure. This was the example that I gave earlier. Pacinian corpuscles will be found much deeper in the dermis. So I just want to take a quick second to review the layers of the skin so that we can visualize where these two receptors will be found. There are 5 layers of the epidermis, and we don't need to go through those 5 layers again. That's something that you would have done in Anatomy and Physiology 1. But I do want to remind you that the dermis is composed up of 2 layers, specifically a papillary layer and a reticular layer. The papillary layer is a bit shorter and contains these epidermal ridges. We call these dermal papillae. And so it is the Meissner corpuscle that will be nestled up here in the dermal papillae. And very deep in the reticular layer is where we will find our Pacinian corpuscles. So the Meissner corpuscle, which is found up here, senses light touch. Therefore, it makes more sense that it's going to be found much more superficial in the dermis, compared to the Pacinian corpuscle, which is going to sense deep touch and found in the deep dermis. This picture will nicely show us the Meissner corpuscles and the Pacinian corpuscles in their respective locations. Because the Meissner corpuscles are found more superficially, up closer to the epidermis, when there's some type of deformation of the skin, that stretch of the skin is going to be sensed by the Meissner corpuscles first. So much lighter touch, such as the touch of a feather, will very, very slightly deform the skin, and that can be sensed by the Meissner corpuscles that are found very close to the epidermis. But in order to stimulate a Pacinian corpuscle, we have to have a much harder, more firmer pressure being exerted on the skin. That is specifically why the Pacinian corpuscles are found much deeper in the dermis. We do have lots of other components that can be found in the skin, but these are the 2 tactile receptors that we want to be focused on in this presentation. One thing that I'd like to mention is that you will have to be able to identify what a Meissner corpuscle and a Pacinian corpuscle look like under the microscope, and you'll have to be able to distinguish them. So on this slide, you can see that the Pacinian corpuscle has many different layers, and this is a very characteristic appearance of the Pacinian corpuscle. Pacinian corpuscles are often described as looking like an onion. So one way that I remember that the Pacinian corpuscle is the one that looks like the onion is I call this the Pacinian corpuscle. That's rather corny if I do say so myself, but it's a little trick that you can use to remember this particular tactile receptor. The Meissner corpuscle, on the other hand, looks a little less organized under the microscope. You can see some histological sections of Meissner corpuscles and Pacinian corpuscles on this slide. The Pacinian corpuscle you can see here with all of its different layers. I remember that since I'm calling this the Pacinian corpuscle, that onions grow deep within the ground, and therefore we can remember that the Pacinian corpuscle will sense deep touch. The Meissner corpuscle, nestled up here in the dermal papillae, will sense light touch, and these are the ones that look a little less organized on the inside. I have personally heard a lot of opinions as to what the Meissner corpuscles look like, but to me, I think the Meissner corpuscle looks like a big bag of ramen noodles. So make sure that you can identify these 2 corpuscles of touch, what they look like under the microscope, and specifically the type of stimuli that they will detect. We're now going to switch gears and start talking a bit more about receptor localization. Brain anatomy would have been covered in Anatomy and Physiology 1 lecture in lab, but I want to take us back here to the brain to talk about two very important structures that we'll see on the brain that are directly related to our body's receptor density. So specifically, on the brain, we have a couple different lobes. Hopefully we can remember the frontal lobe, the parietal lobes, the occipital lobe, and the temporal lobe. But specifically, there is a very, very large sulcus that will separate the frontal lobe from the parietal lobe, and that is known as the central sulcus. You can see this is the central sulcus here, and everybody will have this central sulcus. As a reminder, a sulcus is one of the invaginations of the brain, also referred to as one of the valleys. And gyri are referred to as the hills, or the outward foldings of our brain. So specifically, there are two very important gyri that we need to be aware of for this presentation. Specifically, we can see that this is what's known as the precentral gyrus, and this purple gyrus is known as the postcentral gyrus. We call this the precentral gyrus because it's in front of the central sulcus, and we call this the postcentral gyrus because it is behind the central sulcus. The precentral and postcentral gyri are home to two very important parts of the cerebral cortex, namely the primary motor cortex and the primary somatosensory cortex. Specifically, the primary motor cortex can be found housed within the precentral gyrus, and the primary somatosensory cortex can be found housed within the postcentral gyrus. And we see that shown on this slide. The motor cortex, shown in red here, is also demonstrating the precentral gyrus, and our postcentral gyrus, shown in blue, is going to be demonstrating our somatosensory cortex, and you can see those shown here. So what we have on this slide is a section of the precentral gyrus on the left side, and a section of the postcentral gyrus on the right side. Let us take a quick second to remember that the cerebral cortex is this layer of gray matter that wraps around the cerebrum of the brain. And it is the part of the cerebral cortex that can be found in the precentral gyrus that contains our primary motor cortex, and the parts of the cerebral cortex contained within the postcentral gyrus, which will enclose our somatosensory cortex. So the motor cortex specifically is the part of the brain that helps us to send out motor signals to our muscles. If we are to move our fingers, it is this part of the cerebral cortex that will be active, instead of dealing with sending motor signals out. The primary somatosensory cortex focuses on localizing sensation to different parts of the body. So it is the primary motor cortex that will be in control of sending motor signals out to our muscles, and it is the primary somatosensory cortex which will help us to register sensation across the body. So one thing that you can probably notice about this slide is that we have different parts of the body that have been placed on top of each of these different cortices. What this is indicating to us is that the particular parts of the cerebral cortex that are associated with a particular part of the body are specifically the parts of the brain that control the motor output to that part of the body and the sensation coming back in from the body. Motor signals going out to the body would be efferent signals, and sensory stimuli coming back to the brain would be referring to afferent signals. So now that you can see where each of the body gets its motor innervation and sensation from, I also want to note that the hands and the face are taking up a very, very large amount of the surface area of the brain. And the reason that we are depicting the brain this way is because of the statement shown up here. There are receptors that are located in different concentrations throughout different parts of the body. What this means is that not every area of the body is saturated with the same amount of receptors. Certain areas are going to have more receptors, and that will allow us to use those particular body parts for more fine movements. That's why we see with both of these cortices, the hand and the face taking up the majority of the brain's surface area. This is a very big statement. The more surface area of the brain that's devoted to a particular area of the body, the greater the concentration of the receptors on that part of the body. One example of this is texting. We have a lot of surface area here for very, very fine motor movements with our hands. We can text very, very crisply and quickly with our hands. However, it would be much, much more difficult for us to learn how to text with our knee as well as we do with our hand. This is also shown in the face with facial expressions. Facial expressions are very, very precise motor movements that give us the ability to convey things like feelings to our friends and families. If we did not have as much surface area of the brain devoted to the muscles in the face, we would not be able to make as many precise facial expressions as we are able to. The same thing is true over here with the somatosensory cortex. The hand and the face are areas that are going to be much, much more sensitive to touch. I will use a cell phone as another example. If I was to tell you to close your eyes and I handed you my cell phone, you'd most likely be able to tell me that that is my cell phone just by feeling the smooth surfaces and characteristics of the cell phone. However, if you were to take that cell phone and put it on your leg, it would be much more difficult to determine specifically what object I have given to you. So one of the biggest takeaways from this slide is that the surface area of the brain is directly correlated with the receptor density throughout the body. There is a three-dimensional depiction of receptor density that I want to show you. It is this structure that is called the homunculus. The homunculus just demonstrates what we would look like if our receptor density was correlated with body size. We would have these very, very large hands and a very, very large head as well, since these are the areas that have the greatest density of receptors. So it is the homunculus that is demonstrating receptor density. So there are four remaining concepts that we need to discuss in terms of general senses. You'll find the lab manual sheets for this presentation posted on Canvas, and in the lab manual sheets, you'll find a couple experiments that you can do on your own time to demonstrate these particular concepts. The first concept that we're going to discuss is two-point discrimination. So we've already discussed that our touch receptors are distributed unevenly over the body. The more touch receptors that are in a particular area, the larger the area of the brain that is necessary to process the tactile input coming from that area. Two-point discrimination gives us a way that we can assess our own receptor density, and to do this we'll need either a pair of calipers or scissors. You'll want to make sure that on the device that you are using that the points are rather sharp. So you will find instructions about how to do each of these lab demonstrations in your lab manual sheets, but I am going to walk you through the concepts of each of these and describe how these experiments are done. So in order to find our two-point threshold, we're going to use a pair of calipers or scissors. The two-point threshold can be defined as the smallest distance that you can perceive between 2 points. And this is why we use scissors for this example. When scissors are closed and both of the points are together and we are to touch that to the skin, it feels like it's just 1 point. And so you can slowly open the scissors to find out what your two-point threshold is. So you are more than welcome to pause the video and grab a pair of scissors to work this through with me as I explain it. You want to first take the scissors closed and gently touch the combined point to the ventral forearm. From here, you will gradually increase the distance between the points. And when you feel 2 distinct points, this is going to indicate that you have started stimulating 2 different tactile receptors. At the distance where you start to feel 2 different points instead of 1, you want to measure that distance, and that would be your two-point threshold for that particular part of the body. You'll repeat this experiment on your calf, on your palm, and on your thumb. What you should expect to see is at the areas of your body where there's a denser receptor arrangement, you should find that the two-point threshold is smaller in those areas. The denser the receptors are, the better we can sense and distinguish 2 different points. This is two-point discrimination. The next concept that I want to share with you is tactile localization. Tactile localization can be described as our ability to determine where we have been previously touched. Tactile meaning touch. Tactile localization is our own ability to localize the place that we have been touched. So for this, you'll use 2 different colored pens. So you're welcome to get your little brother or little sister or one of your parents involved in this experiment with you. Essentially, you'll need 2 different colored pens. You will give the first pen to your surrogate lab partner and have them touch you on your ventral forearm. Without looking, you will take the other color and you'll try to touch the exact same place that your surrogate lab partner had initially touched the first time. Once you and your surrogate lab partner have made a dot on your forearm, measure the distance between those 2 points. What we should expect to see is that areas of our body that have a denser receptor arrangement will have a shorter distance between those 2 points, because your ability to reproduce your own somatosensory cortex map should get better as your receptor localization gets denser and denser. So once again, we will see receptor density also playing a big role with tactile localization. Adaptation is the next concept that we're going to cover. Most of our senses do display adaptation except for pain. Pain is not a stimulus that we would want to adapt to. The mechanism of adaptation is still not quite understood. However, we can describe adaptation as the loss of receptor sensitivity due to some type of prolonged stimulus. Touch receptors will adapt to particular stimuli if that stimulus remains long enough and is not particularly painful. Some receptors take longer to adapt than others. The way that we will assess adaptation is by taking some small flat object. A penny or a dime should do just fine. Essentially, with the last two places that you've assessed 2-point discrimination and tactile localization, you will also use those same places to assess adaptation. So you'll take the small coin and place it on your ventral forearm. Initially, you will feel the temperature and the weight of the object, but after a little while, your tactile receptors will adapt, and you'll no longer sense that object there anymore. Adaptation is a very necessary characteristic of our general senses. It is through adaptation that we can focus upon more important stimuli. One example I like to give about adaptation are socks. For those of you that are wearing socks, I'd be willing to bet that until I just mentioned that you are wearing socks, that you are not thinking about wearing socks. So this is a good example for us to imagine adaptation. We would not be a very efficient species if all day we were thinking about wearing socks. We have to be able to sift through the non-important stimuli so that we can focus on stimuli that's more pressing. And I would say that in just about every example, wearing socks is not pertinent information that you need to fill your day with. The last concept that we'll cover here in general senses is referred pain. Referred pain can be described as pain felt in one area that's actually coming from a different area. The brain determines where sensations come from based upon the path that it's taking to get to the brain. Oftentimes, our brain confuses the actual place that is feeling the pain. Internal organs that share a spinal nerve with one part of the skin will often cause pain on that area of the skin instead of in the organ themselves. Referred pain can actually be used medically to diagnose certain conditions based on where the pain is felt. It is much more likely that we are to feel pain on the skin just above the left chest than in the heart. So therefore, with referred pain, our brain is interpreting that pain as coming from the area that is more likely to exhibit pain. The example I just gave is shown here. Pain from the heart can be felt in the chest and all the way down the medial side of the left arm. We will also often find that pain from appendicitis is going to be felt in the umbilical region. The last experiment that you'll find in your lab manual sheet is demonstrating referred pain. Specifically, the ulnar nerve courses along the olecranon process of the elbow. That's what we often refer to as the funny bone. To complete this experiment, you'll need a cold bowl of water. Sticking your elbow into the bowl of water will initially cause some type of cold or tingling sensation. As time progresses, the cold tingling sensations will start to feel rather painful in the posterior side of the last two fingers. Mark the amplitude of the pain for every single minute for 5 minutes. This gives us a way to artificially exhibit referred pain. Of course, I want it to go on record that if the stimulus becomes too painful, pull your arm out of the bowl. Each of these experiments is described in much more detail on the lab manual sheets, so make sure that you print those out and go over those. That will conclude our presentation on general senses. If you have any questions at all, please make sure that you are reaching out to me. I can only be of service to you if I know that you're struggling. We will come back for our next lecture presentation when we will cover the special senses.